Introduction to Food Microbiology. As we all know, food microbiology is also one of the most important branch of our microbiology, and it also has various applications in our day-to-day life. The contents include first and just an introduction, next is about the science of microbiology, next origin and scope of food microbiology, importance of the microorganism in food, and also the food associated microorganisms including bacteria, fungi, yeast and viruses. Next is the factors which affect the growth of microorganism in food items. Next is the significance of food microbiology and the applications of food microbiology. So let's start with an introduction about what is microbiology. What is the significance of the science of microbiology? As we all know what is microbiology and what is the importance of studying microbiology and what are all the importance of the microorganisms, especially in this pandemic season or especially in this COVID scenario. I think there is no need to give much introduction about what is the importance of microorganisms or what is the significance of the science of microbiology. We all know that the entire world is now stuck because of a single microorganism. So that much importance is the science of microbiology. And what is this microbiology? We all know that microbiology is the study of microorganisms, and microorganisms are the one which we cannot see with our naked eye. We require the help of a microscope to observe these microorganisms. And these microorganisms are extremely important in our day-to-day lives. And also some of them are disease causing. And it is also involved the maintenance and modification of our environment and have essential role in industrial field used in the production of different food, beverages and antibiotics, and also body's ability to mount defense against the infectious microorganisms. So microbiology, it is a study of microorganisms, and these microorganisms have application in different areas in our day-to-day life. That is, they are extremely important in our everyday life. Some of these microorganisms are the reason behind different diseases of humans, plants and animals. And also they are involved in the maintenance and modification of our environment. They also play a significant role in our environment maintenance and modification. And besides this, they have significant application, or they are used in the industrial production of various products and mainly used in the production of food items, beverages, antibiotics, vaccines, etc. etc. And also they are involved in the body's ability to mount defense against different infectious microorganisms. Then, what are these microorganisms? We know that they are the living entities which are of microscopic size, and the different microorganisms comes under the group bacteria, viruses, fungi, algae, and protozoa. And we all know that these microorganisms are present everywhere. That is why they have different applications, or they are much important in our day-to-day life. That is, they are present everywhere, and also they have both, and they are both advantages and disadvantages, since some microorganisms are very useful and have many positive applications. At the same time, many of them are pathogenic and also will lead to the various diseases in humans, plants and animals. Next, coming to the origin and scope of the food microbiology, the different topics of major interest in coming under the food microbiology. They have different applications in the field of food fermentation, used for the analysis of water quality, used in the food preservation techniques. They are also the reason behind the spoilage of different food items, and also used in the quality control of food items, and they are used in, and they are the reason behind many foodborne diseases, and also how lab application in laboratory management and food hygiene. So what are the different areas that a food microbiologist have to specialize, or microbiologist must be aware, is that they have different positive and negative areas, or there are many advantages and disadvantages of these microorganisms. They are used in many applications like the production of many food items through the fermentation process, and also used in the quality control of food items, and also the reason behind the many foodborne diseases, foodborne spoilage, food hygiene, food preservation, quality control, etc. etc. Now coming to the various groups of microorganisms and their association with food, we know that all the groups of microorganisms have role in food. That is, even bacteria, fungi, yeast, viruses, everything have both advantages and disadvantages role in concern with the or in association with the food. And coming to bacteria, they are the reason behind many foodborne diseases and food spoilage. At the same time, many species of bacteria are used for the production of many valuable products through the food fermentation, and also used for the production of many food additives and enzymes. So, some group of bacteria will lead to the many foodborne diseases and thereby the spoilage of the food items, and also lead to the wastage of the food items. But at the same time, many valuable products are produced on large scale with the help of some other groups of bacteria. And also the secondary metabolites, so the products produced by some bacteria have application as the food additives and enzymes, so they can be used for the production of many valuable products, and at the same time they will lead to the many food spoilage and foodborne diseases. And then coming to the moles, they are also the reason behind some foodborne diseases and food spoilage. But at the same time, some moles can be used as a single cell proteins, and some moles can be used in the production of many enzymes, food products, and also used in the food fermentation process. Next, coming to the yeast, they are also involved in food spoilage and foodborne illness, and some can be used for in the food fermentation process for the production of valuable products from cheap raw materials, and also some products produced by this yeast can be used as the food additives or the foodborne enzymes. Next is the viruses. They are also the reason behind many foodborne diseases, and they are also used as an indicator for the identification of many food poisoning bacteria, and also they will lead to the failure of many dairy fermentation processes. Next is the importance of microorganisms in relation with the food. First one is, most of these microorganisms are the reason behind many foodborne diseases. Many pathogenic microorganisms, including bacteria, molds, and viruses, can contaminate the food during the various stages of their handling, during the production process, and even during the consumption. And the consumption of this food can lead to many foodborne diseases, and the foodborne diseases can be sometimes fatal and may also cause large economic losses. And the food of animal origin are associated more with foodborne diseases than food of plant origin. First important role, or the first important area is the foodborne diseases. We know that many pathogenic microorganisms, it can be either bacteria, fungus, viruses, they will lead to the contamination of various types of food items. Since these microorganisms are present everywhere, there is equal chance of contamination from the collection of the raw materials up to the preservation or the storage of the food items. These microorganisms are present in the water we are used for preparing the food items. It is present in the soil from where we are collecting the raw materials, and it is also present in the different utensils we are using, and also even in the laborers or everywhere in air, everything in the microorganism is present. So they will lead to the spoilage of the food at any stages. It can be either during the handling process, or during the production process, during the storage, during the transportation, and up to the consumption. There is equal chance of contamination of the food items by these microorganisms. And consumption of these microbially contaminated food will lead to the foodborne diseases. And some of these foodborne diseases are fatal, and it sometimes lead to the death. and some of these foodborne diseases can be fatal and also may lead to large economic losses. The spoilage by the microorganism will lead to the wastage of the food items and thereby large economic losses. And usually the food of animal origin are more associated with the foodborne diseases as compared to the food of plant origin. Next one is the food spoilage. Except the sterile food, all food harbor different kinds of microorganisms, and the food spoilage stems from the growth of these microorganisms in food or is due to the action of different microbial enzymes. And the major concerns are the economic loss and the wastage of the food, and new concepts are being studied to reduce the contamination as well as control the growth of the spoilage microorganism in food. Second one is the food spoilage. We know that the microorganisms are present in almost all food items except in the sterile food. And the food spoilage will starts from the growth of these microorganisms in food, or it may be due to the action of the microbial enzymes. So once these microorganisms enter into a food item, the food spoilage may be either due to the growth of these microorganisms in the food, and it makes unacceptable, convert it into an unacceptable form, or sometimes it will produce some unfavorable enzymes which will lead to some unfavorable changes in the food items and make it unacceptable. And the major concerns are the economic loss and wastage of the food. Obviously, this microbial spoilage or the microbial contamination will lead to the wastage of the food items and will thereby lead to large economic loss. And now new concepts are being studied to reduce the contamination as well as control the growth of spoilage microbes in food. So the researches are going on and many techniques are being developed in order to reduce this microbial contamination and thereby reducing the growth of the spoilage microorganism in food, and thereby help to reduce this economic loss and also the wastage of food items. Third one is the food bioprocessing. Many food grade microorganisms are used to produce different types of fermented food using raw materials from animal and plant sources. And consumption of this food has increased greatly over the last 15 to 20 years and is expected to increase further in the future. There have been great changes in the production and availability of these microorganisms to meet the large demand, and in addition, novel and better strains are being developed by using the genetic engineering techniques. And the third one is the food bioprocessing. And the first two one are the negative impact of the microorganism in association with the food, and this one is the one of the positive impact of microorganism, that is they are mainly used in the food bioprocessing. Many food grade microorganisms are used to produce different types of fermented food using raw materials from the animal and plant sources. So one of the important application of this microorganism is that they can be used to produce many valuable products on large scale with the help of the microorganism. These microorganisms help to convert cheap raw materials from the animal and plant sources to valuable products. And the consumption of such food has increased greatly over the last few years and also is expected to increase further in future. So day by day the demand of such valuable products is increasing, and there have been great change in the production and availability of these microorganisms in order to meet this large demand. And in addition, novel and better strains are being developed by using the genetic engineering techniques, and day by day many techniques are being developed in order to improve the activity or improve the production ability of this microorganism. And also we are using the genetic engineering techniques in order to develop new strains which have the ability to produce new valuable products, and by developing such new strains we can easily convert the cheaply available raw materials into valuable products on large scale with the help of these microorganisms. And the fourth one is the food additives. Many microbial enzymes are used to produce food and food additives, and by employing genetic recombination techniques and by using diverse microbial sources, enzymes of higher purity and activity are obtained. And many types of additives from microbial sources are being developed and used in food. It includes essential amino acids, color compounds, flavor compounds, stabilizers, organic acids, etc. Another positive impact is the use of the food additives. Many microbial enzymes are used to produce many food and food additives. Many microbially produced compounds can be used as the food additives, and by employing the genetic recombination techniques and by using the diverse microbial source, enzymes of higher purity and activity can be produced or can be obtained. And many types of additives from microbial sources are developed and used in food, and instead of these chemical preservatives or chemical additives, we can use this microbially produced additives. And if some examples are some essential amino acids, color compounds, flavor compounds, stabilizers, and organic acid. So such compounds which are produced by the microorganisms can be used as a additive, can be used as the food additive in order to improve the keeping quality of the food items or improve the taste of the food item, improve the appearance of the food item. So additives are anything we are adding to the food in order to increase its acceptability, that is anything we are adding to the food other than its normal constituents comes under the group of food additives. So many microbially derived products can be used as the food additives. It is also one of the important advantage of these microorganisms. So in the development, large scale production of these microbially derived food additives will help to reduce the use of chemical food additives. Next one is the food biopreservation. Some antimicrobial metabolites like the bacteriocins and organic acids like acetic acid, propionic acid, and lactic acids of desirable microorganisms are being developed and used in food in place of preservatives in order to control the pathogenic and spoilage microorganisms in food. Economic production of these antimicrobial compounds and their effectiveness in food systems have generated wide interest. And the fifth one is the food biopreservation. We know that some microorganisms can produce some metabolites which act as an inhibitory factor against, say, in some other pathogenic microorganisms. So such antimicrobial metabolites examples are the bacteriocins, acetic acid, propionic acid, and lactic acid, which are produced by these desirable microorganisms can be used to reduce the growth of some other food spoilage microorganism, and thereby these microbially produced metabolites can be used as a preservative. Hence, these microbial products can help to reduce the use of the chemical preservatives in food items. Instead of the chemical preservatives, we can add these microbially produced preservatives to the food items and thereby help to increase the shelf life of the food items. And this helps to control the infection from the pathogenic and spoilage microorganisms in food. And the economic production of these antimicrobial compounds and their effectiveness in food system have generated now wide interest. Next is the uses of probiotics. Probiotics are the live microorganisms which are used as food and feed supplement. And WHO defines probiotics as the live microorganism which when administered in adequate amount will confer a health benefit to the host. And they act generally by improving or restoring the gut flora, mainly bacteria, but some yeast can also used as the probiotics, and the broad spectrum probiotics or multi probiotics will combine different species in the same product. Probiotic food include yogurt, kefir, sauerkraut, timber, kimchi, etc. And the next is use is the probiotics. Probiotic, we know that microorganisms itself can also be used as a food and feed supplement, and these probiotics are the live microorganisms which can be used as a food and feed supplement. And according to WHO, probiotics are the live microorganism which when administered in adequate amount confer a health benefit on the host. So these microorganisms, when consumed in adequate amount, will be beneficial to the host, that it will help to improve the health wise characters of that host, and it also act generally by improving or restoring the gut flora. And the significance of this probiotic microorganism is that it helps to improve or restore the gut flora. That is, there are some favorable or there are some advantages microbial load in our gut, and which will help in our digestive functions. And with the help of this probiotic, or the consumption of these probiotics, will help to improve or restore the gut flora. And mainly bacteria, yeast, and fungi can be used as the probiotics. And what are the broad spectrum probiotics or multi probiotics? Are they are the combined combination of the different species in the same products. That is, broad spectrum and probiotics or multi probiotics are different species of the microorganisms which can be used as a probiotics. And some probiotic examples of some probiotic food are the yogurt, kefir, yogurt, tempeh, kimchi, etc. These are the food which will help to improve the health-wise benefits of the host, and consumption of this food is very much beneficiary to the host. Next is, what are the common types of microorganisms which are associated in food. First one is the bacteria. We all know that bacteria is more abundant in nature, and also they are more commonly associated with the food as compared to other groups of microorganisms. And among the bacteria, the first group is the lactic acid bacteria. These are the bacteria that will produce relatively large quantities of the lactic acid from carbohydrates, and it include the species mainly from Lactococcus, Leuconostoc, Pediococcus, Lactobacillus, Streptococcus, Thermophilus, etc. So the first group of bacteria that are commonly associated with the food is the lactic acid bacteria. They are the group of bacteria that have the ability to produce large quantities of the lactic acid from carbohydrates. Common examples are the Lactococcus, Leuconostoc, Lactobacillus, etc. And the second group is the acetic acid bacteria, and these are the group of bacteria that have the ability to produce acetic acid. And one example is the Acetobacter aceti. Third one is the propionic acid bacteria, and these are the group of bacteria that will produce propionic acid, and they are mainly used in the dairy fermentation. They are mainly used in the production of the many dairy products. And one example is a Propionibacterium frudentrici. Next group of bacteria is the butyric acid bacteria, and these are the group of bacteria that are able to produce butyric acid in relatively large amount. And example is the Clostridium butyricum. Next group is the proteolytic bacteria, and these are the group of bacteria that are capable of hydrolyzing the proteins and due to the production of some extracellular proteinases. So the proteolytic bacteria are the group of bacteria that have the ability to hydrolyze the proteins present in the food items, and it is mainly due to the production of some extracellular proteinases by these bacteria. Examples are the Micrococcus, Staphylococcus, Bacillus, Clostridium, Pseudomonas, etc. And the next group is the lipolytic bacteria. They are able to hydrolyze the triglycerides, and it is mainly due to the production of some extracellular lipases by these microorganisms. Examples are the Micrococcus, Staphylococcus, Pseudomonas, etc. Next group is the thermoduric bacteria, and these are the group of bacteria that are able to survive pasteurization temperature. We know that pasteurization is the technique we are used to for the preservation of milk and milk related products, and these thermoduric bacteria are the one that will lead to the contamination or spoilage of milk and milk products, because they have the ability to survive the pasteurization temperature. Examples are the Micrococcus and the Enterococcus. Next one is the halotolerant bacteria, and they are able to survive high salt concentration. Examples are the Bacillus and the Staphylococcus halotolerant. They are the group which comes under, which have the ability to survive under high salt concentration. We know that salt solution is one of the simplest or commonly used method for the preservation of many fruits and vegetables, meat, fish, etc. And this halotolerant bacteria, the group that will lead to the spoilage, or that will cause the spoilage of such food items, because they have the ability to survive under high salt concentration. Two common examples are the Bacillus and the Staphylococcus. Next group is the acidic bacteria, and they are able to survive at low pH. That is, they are able to survive in acidic condition. They are able to survive in a pH below 4. Examples are the Lactobacillus and the Pediococcus. So these acidic bacteria are the one which will lead to the spoilage of the food items which contains high acidic acid content. That is, these are one which will we can see in pickles, etc. And these acidic bacteria have the ability to survive at a low pH, that is pH below 4. And examples are the Lactobacillus and Pediococcus. Next is the gas producing bacteria. These are the group of bacteria that have the ability to produce the gases like carbon dioxide, hydrogen sulfide, etc. during the metabolism of different nutrients present in the food items. Examples are the leuconostocs, lactobacillus, escherichia, etc. So gas producing bacteria are a group of bacteria that have the ability to metabolize the components of the food items or the nutrients of the food items with the liberation of some gases like carbon dioxide, hydrogen sulfide, etc. So if a food item is contaminated or spoiled by this group of microorganisms, the symptom is the formation of bubbles on the surface of the food item. And the examples are the leuconostocs, lactobacillus, escherichia, etc. And the next group is the slime producers. They have the ability to produce slime due to the synthesis of polysaccharides. So slime producers are a group of bacteria that are able to produce or synthesize polysaccharides and will lead to the formation of a slime on the surface of the food items, and it includes some species like Xanthomonas, Leuconostoc, etc. Next one of the more pathogenic group is the spore formers, because they have the ability to produce spores and they able to survive under adverse conditions. So they are able to survive high heat, low temperature, etc. So they are able to survive most common preservation techniques. So these spore formers are most important group which are associated with many foodborne diseases. Examples are the Bacillus clostridium species. Next group is the aerobes. As we all know, aerobes are the group of bacteria that require oxygen for their normal growth and multiplication. Examples are Pseudomonas, Bacillus, Flavobacterium, etc. And obviously, anaerobes are the one that cannot survive in the presence of oxygen. Example is a Clostridium species. Next is the facultative anaerobes. These are the group of bacteria that are able to grow both in presence and absence of oxygen. Example Lactobacillus, Pediococcus, Leuconostoc, etc. So aerobes, anaerobes, and facultative anaerobes are the three groups of bacteria in association with or in connection with the oxygen requirement. Aerobes requires oxygen for their growth and multiplication, whereas anaerobes can survive under anaerobic condition. They can grow in the absence of oxygen, and facultative anaerobes are another group that can survive both in presence and absence of the oxygen. Next one is the coliforms. They include mainly species of Escherichia, Enterobacter, Klebsiella, etc. And it is also one used as an index of sanitation. The presence of presence of coliforms in water or in food items may is an inference about the unhygienic practices followed during the production of such food items. Next is the fecal coliforms. It also include E. coli, and it is also used as an index of sanitation. If the presence of coliforms in water or food is an indication of the fecal contamination in such food items. Next is the enteric pathogens. They include pathogenic Salmonella, Shigella, Campylobacter, Yersinia, Escherichia, Vibrio, etc. And these are the one that will lead to many gastrointestinal infections. Then coming to the major food associated fungi, first one is the aspergillus. They are widely distributed and contain many species that are important in food. And many of them are xerophilic, that is, they are able to grow in low water activity and can even grow in grains and lead to the spoilage of such food items. And the spoilage of food such as jams, nuts, fruits, and vegetables are also mainly caused by this aspergillus species, and some species will produce a toxin, that is a fungus produced toxin is the mycotoxin. Example is the aflatoxin, which is produced by the aspergillus flavus, and many species or strains of aspergillus are also used in food and food additive processing. So the first group of fungus is the aspergillus. They are widely distributed and contain many species that are important in relation with food, and many of them are able to grow in low water activity. They can survive in low moisture content, and also they will lead to the spoilage of grains and jams, nuts, fruits, vegetables, etc. And another important factor is that they will produce some mycotoxin. Fungal producing toxin is a mycotoxin, and one example is the aflatoxin, which is produced by the aspergillus flavus. But at the same time, many species of the aspergillus are also used in the food and food additive processing. Second group is the Alternaria. They are also septate and form dark brown colored conidia on the conidiophore. They cause rot in tomatoes and produce a rancid flavor in dairy products. Second group is the Alternaria, and coming to their structure, they are septate and form dark brown colored conidia on a conidiophore. And coming to the diseases, they will cause rot in tomatoes and also produce a rancid flavor in many dairy products. And the third one is the geotrichum. Here also the hyphae are septate, and also they form some rectangular asexual arthrospores, and they establish easily in the equipments and often grow on many dairy products, and since they are also known as the dairy mold. So which one is the dairy mold? Geotrichum is the dairy mold. Why this geotrichum is known as a dairy mold? Because they can easily establish in equipments. They can easily grow in equipments and often lead to the spoilage of many dairy products, and hence they are known as a dairy mold. Next one is the mucor. They also have non-septate hyphae and also produce sporangiophores. And some species are mainly used in the food fermentation and also in the production of many valuable enzymes. At the same time, they will also lead to the spoilage of many vegetables. So the fourth one is the mucor. They have non-septate hyphae and also produces sporangiophores, and some species are used in the fermentation of valuable products and also used in the production of many applicable enzymes. At the same time, some will lead to the spoilage of many vegetables. Next is the penicillium. They have septate hyphae and form conidiophore on a blue-green brush-like conidial head, and subspecies are used in the food production. Examples are the Penicillium roqueforti and Penicillium camembert, which is used in the cheese production. So the next one is the penicillium. They have a septate hyphae and form conidiophores on a blue-green brush-like conidial head, and some species are used in the production of valuable products. Example is the Penicillium roqueforti and Penicillium camembert used in cheese production. But at the same time, some species of this penicillium will also lead to fungal rot in fruits and vegetables. Next one is the rhizopus. Here the hyphae are aseptate and also form sporangiophores in sporangium. They are involved in the spoilage of many fruits and vegetables. Rhizopus stolonifer is the common black bread mold. Next one is the rhizopus. Its hyphae are aseptate and also form sporangiophores in sporangium. They are also involved in the spoilage of many fruits and vegetables. And the Rhizopus stolonifer is the common black bread mold. Coming to the different yeast species associated with food, first one is the Saccharomyces. Here the cells are round, oval or elongated, and it is the most important genus and contains heterogeneous groups. Saccharomyces cerevisiae variants are used in baking, for leavening of bread, and in alcoholic fermentation. They are also involved in the spoilage of food with the production of alcohol and carbon dioxide. So the next group is the yeast. First one is the Saccharomyces. By structure, they can be round, oval, or elongated, and it is the one of the most important genes or genus, and also contains heterogeneous groups. And the Saccharomyces cerevisiae is mainly used in the leavening of bread and also in many alcoholic fermentation process. And at the same time, many species of Saccharomyces are also involved in the spoilage of many food items with the production of alcohol and carbon dioxide. Next one is the Rhodotorula. They are pigmented red, pink, or yellow and form yeast and can cause discoloration of food such as in meat, fish, sauerkraut, etc. One example is the Rhodotorula glutenis. So next one is the Rhodotorula. They are the pigmented forms. They are sometimes red colored, pink colored, or yellow colored, and will lead to the discoloration of the food. When once this Rhodotorula infect a food item, it is easily visible by the color change of the food, that is a discoloration in meat, discoloration in fish, discoloration of sauerkraut, etc. One example is the Rhodotorula glutenis. Next one is the Torulopsis. They are spherical to oval in structure, and they will lead to the spoilage of milk due to their ability to ferment lactose. Example is the Torulopsis spherica, and they also spoil fruit juice concentrates and some acid foods. Next one is a Taurolopsis, and they have spherical or oval structure, and they are the reason behind the spoilage of milk, and it is mainly due to their ability to ferment lactose. Example is a Taurolopsis spherica, and besides milk, they will also lead to the spoilage or the fruit juice concentrates and some acidic food items. Next one is a Candida. Many of the Candida species will spoil food with high acid, salt, and sugar, and also form pellicle on the surface of the liquid. And some can cause rancidity in butter and dairy products. Example is the Candida lipolytica. Next one is a Candida. Many Candida species are able to grow or survive in high acid, salt, and sugar food items, and they will lead to the formation of a pellicle on the surface of the liquids, and some will even lead to the rancidity of butter and dairy products. One common example is the Candida lipolytica. We know that salt solution, sugar solution, etc. are the common preservation solution or preservative techniques we are used for the preservation of many food items, but this Candida species can survive in this high acid, salt, and sugar solutions, and due to the spoilage of such food items. The spoilage can be indicated by the formation of a pellicle on the surface of the liquids. Besides that, they will lead to the spoilage of butter and dairy products. Example is the Candida lipolytica. Next one is the Saccharomyces. They are involved in the spoilage of foods containing high sugar or salt levels. They can grow in honey syrups, molasses, soy sauces, etc. Example is the Saccharomyces nusbemeri, and they can grow in high concentration of these solutes. And next one is the Saccharomyces, which are involved in the spoilage of the food containing high sugar level, high salt level, etc. They can survive. We know that usually honey is used as a preserving liquid for the preservation of fruits, vegetables, etc. And some species of Saccharomyces can even survive in such solutions, and they can grow in high concentration of the solute. Then coming to the viruses, hepatitis A and Norwalk viruses have been the reason behind many foodborne outbreaks, and several other endemic viruses such as the polio virus, echovirus, and Coxsackie viruses have the potential of causing many foodborne diseases. So viruses are also associated with the spoilage of many food items. Many zoonotic diseases are also caused by many viruses. Next is the different factors that affect the growth of microorganisms in food. So that certain intrinsic and extrinsic factors influences the growth and survival of a microorganism in the food. some intrinsic factors or internal factors like the pH, water activity, redox potential, composition of the nutrients, inhibitory substances, and biological structures influences the further growth and survival of a microorganism inside the food. So once these microorganisms enter into a food item, some factors or some parameters or some internal and external factors influences the further growth, survival, and thereby the spoilage of the food by such microorganisms. Intrinsic factors or the internal factors include the pH, water activity, redox potential, composition of the nutrients, inhibitory substances, and biological structures. We know that pH is one of the important factor that require the growth of a microorganisms, that influence the growth of a microorganisms. We know that the microorganisms are highly optimistic in their pH requirement, and hence the pH of the food item is also one important factor that influences the further growth, multiplication, survival, etc. of the microorganisms inside the food. Once they can survive, only they can lead to the spoilage of such food item. Next is the water activity of the food item is also another important factor. Next is the oxidation reduction potential of the redox potential, and also another important factor is the composition of the nutrients. So the nutrient content of the food item is also an important factor that influences the growth of microorganisms in food item. Next, the presence of some inhibitory substances. Sometimes some microorganisms will already present in the food items and they will produce some inhibitory substances. So such inhibitory substances is also an important factor. Next is the biological structures. Mostly the foods have some external layer, and the skin of the meat, etc. Others some common shell of the egg, etc. Some common biological structures present in the food items which will interfere in the entrance of the microorganism to the food items. So these are all the internal or the intrinsic factors that influences the entrance or the growth and survival of a microorganism to a food item. Coming to the extrinsic factors, that include the relative humidity, temperature, atmospheric gases, and also the presence of other microbial flora. So the moisture content of the atmosphere or the relative humidity is also an important factor, because the change in relative humidity will adversely affect the water content and the pH of the food items, and thereby hence this relative humidity or the moisture content of the atmosphere is also one of the external factor that influences the growth of a microorganism in food. And another most important factor is the temperature. Microorganisms are highly optimistic in their temperature requirement. They require a high, they require an optimal temperature for their normal growth and metabolism. For sense the temperature or the temperature of the surrounding of the food is also an important factor. And next is the presence of atmospheric gases. If it is an aerobic microorganism, then it requires the presence of oxygen, and if it is an anaerobic organism, if oxygen is present in the atmosphere, it will inhibit that particular microorganism. And also presence of other gases in the environment is also an important extrinsic factor. Next, another important factor is the presence of the other microbial flora in the environment. If there is any inhibitory microorganisms present in the environment, it will obviously affect the growth and survival of the pathogenic microorganism inside the food item. Then coming to the significance of the science of food microbiology. First one is that they are involved in the foodborne diseases of the microbial origin. Next, they are involved in microbial food spoilage. They have some beneficial uses of microbes in relation with food. They help to control the microbial growth in food. They are used in the destruction of microorganisms in food. They can be used in microbial food fermentation. They can be used as probiotic bacteria, regulatory aspects to ensure the consumers related to the microbial hazards in food. So the different significance, some microorganisms are involved in some foodborne diseases, some are involved in food spoilage, and at the same day there are many beneficial aspects of this microorganism in relation with food. And some microorganisms can also be used to control the growth of some harmful microorganisms in food, and they can be used for the destruction of some harmful or pathogenic microbes in food. they have positive role or positive as use in food fermentation. They can be used as a food or a food feed supplement, that is they can be used as probiotics, and also have regulatory aspects to ensure the consumers related to microbial hazards, because they are also involved in the quality control. The microbial quality of a food is also one important factor that influences the acceptability of a food item. Then coming to the applications of the science of food microbiology, it provides us with the information about the different types of microorganisms. Next is microorganisms used as food. Microorganisms are used in the production of large number of fermented food. Microbiology is very useful in the preservation of the food items. They are involved in many microbial diseases, use of industrial microbiology, and energy from microbial sources. What are the different areas of interest coming under the science of food microbiology, or what are the applications of food microbiology, or what are the areas where we can specialize after studying the food microbiology? First one is that one important application or one important significance is that it provides us a good information about the different types of microorganisms. In addition to getting a job or in addition to study, it will also help to improve our day-to-day lives. We must get an awareness about the significance of microorganisms in relation with food. It will help us to improve our health. It will help to improve our eating habits, and it will also help us to avoid unwanted food items. Like that, it is very much useful, or it provides us information about different types of microorganisms in relation to food item. And also we can easily identify the spoilage of the food items by seeing the gas production, or a color variation, or a slime on the slime formation on the surface of the food item. We get an idea, though it is contaminated with the microorganisms, so it is not useful, or we cannot consume that. So by studying the field of food microbiology, studying the science of food microbiology, it will provide us information about almost all the microorganisms that come in relation with the food item. Next important application is that it gives us an idea that microorganism can be used as a food or feed supplement. It is also another good area, that is it can be used, microorganism itself can be used as a food or feed supplement. Next is the microorganisms are used in the production of many fermented food. It is to help us to utilize, or it help to explore the cheap raw materials for the production of many valuable products in large scale with the help of the microorganisms. After studying food microbiology, we will get an idea, if we are using this microorganism, we can convert those raw materials into a valuable product in large scale, and these are the conditions which we have to provide to such microorganisms. So it will also help us to give an idea about the use of microorganisms in the fermentation of many valuable products. And also microorganisms can also be used in the preservation. We have already told that microbially derived products can be used as a preservative for the long-term storage of the food items. So that is also another important and positive impact. Next is the microbial diseases. We will get an idea about many microbial. It must be the diseases of the plants, diseases of the animals, or foodborne diseases in humans. Everything we will get an idea from this food microbiology. Next is the industrial application. We have already told that these microorganisms can be used in the production of many valuable products on an industrial scale. So with the help of this food microbiology, we can produce many valuable products on a large scale or in an industrial scale. Next is the sources. A microorganism itself can be used as an energy source. Thank you.